Okay, thank you for inviting me here. And let's spend the next 15 minutes discussing about the oncology precision medicine. And I really want to give you here the, let's say, industry perspective here. So why oncology? So the cancer mortality is still one of the main causes of the death, not only in the US, but also in the worldwide. And actually the cancer incidence and cancer mortality are going to increase dramatically in the coming years. Of course, one of the reasons are the aging patient population here, but also it's clearly showing that our treatments are not effective enough. So there's still a lot to be done. If you think about the cancer care here, so there has been traditionally 3 major pillars here. I don't know how this works, but surgery, radiotherapy, and chemotherapy. And recently 2 new pillars were added here, so-called precision therapy and immunotherapy. And the precision therapy is sometimes called like a targeted therapies, and here the examples are like TKI, so there's drugs which are preventing the angiogenesis in a cancer cells. And in immunotherapy, it started already earlier. It's about the antibodies and antibody drug conjugates, and nowadays it's a lot of discussion about the checkpoint inhibitors. So what do we, okay, so sorry about that. So what do we mean about, let's say, precision therapies? So this slide actually shows how we define the increase in precision of the cancer care. Here at the right-hand side, or left-hand side, right-hand side for you, we can see, let's say, the medications which are the least precise. So these are the agents which are damaging the DNA, and they are really targeting both the cancer cells and also the normal cells, not very precise at all. And here in the upper right-hand corner, you can see the medication which has a very high precision. And here we are targeting, let's say, the genetic mutations within the tumor cells. And in the between here, so let's say, and with these, let's say, very targeted drugs, we should see the highest, let's say, efficacy and hopefully the least side effects for the patients. And in the middle, you can see many different treatments like, let's say, radiotherapies and angiogenesis inhibitors and so forth. And if you look at, let's say, our company pipeline here with the green bars, you can see our, let's say, pipeline for the oncology medication. It really covers a lot of those precision principles shown before. So if you look at the late phase development here, phase 3, we have, let's say, here the angiogenesis inhibitors, is this really working? And the radiotherapies. And if you look at the phase 2 here, we are getting more and more precise here. We have our, let's say, immunotherapies here. We have the antibody drug conjugates, which are really targeting very specific proteins on the surface of the cancer cells. And in the further, if we go down with a very early development, we have here the drugs which are really targeting the specific mutations within the cancer cells, like the mutant IDH1 inhibitors. So we are really getting more and more precise in our development as well. So why are we needing this kind of precision medicine? Why do we need these kind of targeted therapies? So if you look at here, let's say the current approach is more like one treatment or one size fits for all approach. So there we give the same treatment for every patient, and unfortunately only, let's say, some of the patients we can see the treatment effect, some of the patients we don't see anything, and then we have some of the patients where we only can see the adverse effects. And if you look at, let's say, the personalized medicine, the future, so actually our aim is to give the medication to the patient according to their molecular makeup. And how do we define the molecular makeup then? So we need so-called biomarker diagnostics. So the biomarkers are reflecting the molecular changes which are driving the patient's cancer. And these kind of biomarkers can be diagnosed from blood, DNA, urine, or tissue samples. And then finally, the patient can be categorized based on their biomarkers in the smaller groups. And then we can give the right medication at the right time to the right patient. And we should see the positive treatment effects for every patient. So are we making progress if we think about the overall 5-year survival rates due the past 40 years? If you look at the lower part of the slide here, definitely no. If you look at the lung cancer and pancreatic cancer, we really haven't made any progress here. Of course, one reason might be that when we diagnose those cancers, they are already at the late stage, they are already metastasized. But if you think about cervical cancers, there we have a very good screening programs, and still we are not making any progress. But if we are looking at the upper part of the slide, yes, definitely we are making some progress here. If you look at overall all the sites, yes, some progress here. And especially if you look at, let's say, leukemia or liver, in leukemia, we have been able to double the 5 years overall survival rates. during the past 40 years from 30% to the 60%, and of course have made a great, let's say, progress in liver cancer and also in the breast cancer as well. So if you think about how we, let's say, traditionally develop the new drugs, we are not very effective there. If I may say so, that less than 1% of the molecules from the very beginning, when we start testing the compounds, will end up, let's say, to the patients and we get the market authorization approval. It's a very lengthy process, takes more than 10 years and costs a lot of money. So how can we improve this? And actually the answer is the same than for the patients with the biomarkers. So finding and designing a biomarker-based drug development program actually has improved our success rates from phase 1 to approval threefold. So it has been less than 10%, and nowadays it's more than 26%. And actually the critical change and improvement has been here in the transition from phase 2 to phase 3, where we have been able, with the biomarkers, able to, let's say, double our success rates. And also the biomarkers actually allow us to develop very novel trial designs. And here are a few examples here, so-called basket trials and umbrella trials. And the basket trials, we are actually targeting the same driver mutation regardless of the underlying diseases. So it might be the mutation, let's say the mutated IDHL in, let's say, AML and glioblastoma, and we put all the patients on the same trial. So actually, this we are already doing quite a lot. But if you think about the umbrella trials here, actually here we are targeting several driving mutations in one specific cancer. Let's take the lung cancer and give, let's say, several treatments to the patient. And this is a little bit more complicated approach since we have to very much collaborate across the academic research organization, also across the pharma companies. And we have some programs here like NCI match programs, as this is also already happening. So how do we tackle as our company about this need to have these precise medication targeted therapies for the patient? So actually this is really a joint effort, and we are very much collaborating in, let's say, in two different components. First, finding, let's say, the new novel targets to be treated and, let's say, the new medication, and also finding and creating, let's say, the so-called companion diagnostic. And this is companion diagnostic is the diagnostic where we diagnose, let's say, where we can diagnose these biomarkers. And actually, if you look at the research part here, I'm really pleased to say that we are, let's say, collaborating quite a lot with the Finnish, let's say, biobanks and companies here, like, let's say, I can see Pharmatest here and Ori and so forth. So we are collaborating a lot here in Finland, and we would like to do even more in the future. And if you think about the companion diagnostic here, so actually here at Bayer, for every new oncology drug, we also have this companion diagnostic development. And actually we have very established partnership for, let's say, these more major technologies like immunohistochemistry with Ventana, and this we are using for our antibody drug conjugates. But we are also trying to tackle, let's say, these cutting-edge technologies here, like a next-generation sequencing and circulating tumor cells. And again, we are establishing here the partnership with the different companies. And again, this is the area we would like to see more collaboration, and also some companies from Finland as well. And this kind of development of the companion diagnostic, it's a long process. We have to start very early at the research. And it starts here with finding the biomarker with the predictive value. Then we have to find the right platform. We have to find the right partner. We have to test, validate that. And actually, it's getting very complicated here when we run the clinical trial. So when we do this kind of targeted recruitment based on the specific mutation, so it might be that this mutation is existing only 1% of the specific cancer patients. So we have to somehow screen maybe thousands of patients in order to treat 20 patients. So it's a lot of effort here, and that's a lot of complication. And also when we are looking for the regulatory approval, it's not enough that we submit the data from our drug. We have to submit the data from the companion diagnostic as well. So it's this kind of tandem approach what we have to do in the future, and of course finally enter the market. And it's very, let's say, challenging for us as a pharma company, and I guess even more challenging for the clinics and also for the patients. So, and this is one of the case examples here. So I mentioned the biomarkers, and then I mentioned the biopsy. So tissues are one of the ways to diagnose these biomarkers. But sometimes when we do this research, the biopsies are not available. Maybe these let's say we don't have slides anymore available from the original tumor, and it's not convenient for the patient to get the biopsies from the distant metastases. So then one answer here is maybe the circulating tumor DNA cells. And then we have to analyze, do the molecular profiling. Sometimes the traditional techniques are not sensitive enough. So do we have a place where we can use these more advanced technologies? Finally, we can identify the right target, which is driving the mutation, driving the cancer. And then there's another challenge here. Where do we get the treatments here? And this comes, let's say, the collaboration. And it's not the thing that we do this circle only once. We have to repeat this several times. Since sometimes the nature is clever. Sometimes, let's say, you get the resistance for the treatments, and then you have to figure out what is driving the more, let's say, relapsed tumor. So have we made any progress, let's say, in the precision medication in the past years? So if you look at the data from 2005 and 2009, it's about 10 years when these new, let's say, precision therapies were introduced. And okay, of course, the studies using this kind of precision medicine approach, they have increased during those years, but still it's less than 10% of the studies which are using this approach. And also, if you look at that they are also targeting these well-validated alterations. So we are not really targeting anything novel so much. So there is still a lot to be done together. And if you look at here, I mentioned, let's say, the biomarkers, and I mentioned the biopsies here. And actually, maybe one of the limitations why it has taken so long time to develop this precision medicine, it's quite challenging to collect the biopsies. And this just shows here that, of course, the number of the biopsies are increasing in a phase 1 studies. But if you look at the data here, that normally in, let's say, traditional phase 1 study, we had something about 20 to 40 patients there. And if you think about that, we collect on average 20 biopsies. It means that we get the biopsies about half of the patients, and we don't even get the paired biopsies. So this is something, let's say, a challenging area where we really have to collaborate together. And maybe one of the answers is here the novel techniques for the circulating tumor cells. And how we are addressing, let's say, the need to get the biopsies in the phase 1 studies at Bayer is our very good network with our external partners. And Bayer is a truly international company. And this slide shows our, let's say, established phase 1 site. So we have a lot of collaboration in US, but it's not limited to the US only. So we are also working very closely, and very many hospitals here in Europe like to see more dots here in Finland and also in Asia. So we are doing a lot of collaboration. And as I mentioned earlier, so this developing the precision medicine is really a joint effort. And actually, I really encourage you to visit our innovation platform called innovation.bayer.com. And this is only the glimpse here. So this is the area where you can, let's say, provide and send us your ideas. We evaluate them, we come back to you. And I really hope that in the future we can work together. I think together we can really advance these precision medicines for the cancer patients who really need these new medications for their advanced diseases. And that's about that. Thank you.